All right. So I thought before I start sort of the main sort of scientific presentation of my talk, it would be worthwhile for me to sort of review my trajectory as a physician scientist to share with you, maybe helpful to some of the trainees, young faculty members, just to sort of show you sort of what my trajectory has been, so that you don't think that it's all sort of hunky-dory, and I think it's helpful to some of the younger individuals who are in the field. So I started my medical school, as Isaac said, at the University of Southern California in Los Angeles. And then I had the pleasure of spending 1 year in the lab with Dr. Peter Jones at USC. So I took a year off between second and third year in medical school to do research. It wasn't very common in those days to take a year off and do research in medical school. Now it's very common. I think it's more than 50% of any graduating medical school class does something different, but it was extremely uncommon in the late 1980s to do that. After graduating, I did my residency training at the Brigham and Women's Hospital. I had the opportunity to go to UCSF for 3 years. It was actually a research fellowship in molecular medicine training program, and then come back to finish my residency training at the Brigham and the Harvard Longwood program in year 2000. And then I started my faculty position at Beth Deaconess Medical Center. Initially, that was my first faculty position. And I was fortunate to have the support of another physician scientist, Dr. William DeWolf, who was the chief of the group at the time. And he himself was a surgeon scientist and had all of the appreciations of the challenges of going through, sort of developing a career that way. And after 7 years of being there, I was recruited to Mass General Hospital. And again, I had the support of Dr. Scott McDougall, another sort of surgeon scientist who's well known in the field. So I was very fortunate to have the support of these individuals that helped sort of foster my career early on, which was extremely important to me in sort of where I am today. And then subsequently, after 11 years of being at MGH, I was sort of had the opportunity to come back to Beth Deaconess Medical Center and start a brand new residency program separate from the Harvard Longwood program. So that was very exciting to me. And I want to sort of highlight sort of the time, actually, the 1 year that I spent at medical school. That 1 year, you'll see still it's that year that I spent at medical school that still sort of is the cornerstone of my research today and all the grants that I have and how that has sort of fostered my career. So this is a publication from the 1980s from the lab that I spent time in, the 1 year that I spent time in. I had no idea that these cytidine analogs and DNA methylation will have anything to do with my career moving forward. And this was one of the sort of the early times when it was appreciated that epigenetic regulation or modification of the DNA, not at the genetic level, but at the epigenetic level, like methylation of the cytidine and pyrimidines, could actually affect cancer progression, development, and so forth. And I must say, at the time, I had zero appreciation for it. But this has become one of the sort of the epic findings of the 21st century for scientific discoveries of how cells and genes are regulated. And then in the year that I spent in the lab, I had the opportunity to participate in this project where methylation of cytosine, we showed that it's important for mutagenesis within the cell. So if you have the methylation of the cytosine with epigenetic modification, the cell has a higher chance of developing spontaneous mutations, both in sort of malignant cells and also in benign cells. And then in the year that I spent there, we also looked at genetic modifications in bladder cancer in a paper that we published and looking at low grade versus high grade. This was the time in restricted fragment of polymorphism RFLP studies, which is very early on, and all of these sort of experiences sort of developed my scientific sort of background discovery and the teaching and the learning that I had in sort of allowing me to sort of pursue a scientific career today. And then, as I mentioned, I went to UCSF for 3 years to do the molecular medicine training program. And one of the papers that we published was one of the first that showed that normal appearing cells, normal fibroblasts associated with cancer cells, known as carcinoma-associated fibroblasts, actually have a significant role in promoting cancer progression. So on the left image, you see how an immortalized cell with human carcinoma-associated fibroblasts promotes tumor growth or progression, whereas if you take normal fibroblasts from areas where there's no cancer associated with it and sort of mix it with the same cells on the right side, there's no significant tumor growth. So this was, again, another sort of level of sort of how epigenetics and things besides the actual appearance and pyrimidine genetic code can affect cancer progression, which was important for my learning of my scientific discovery. So when I started my first faculty position at Beth Deaconess Medical Center, the first funding opportunities that I had, I had some seed funding from the institution. I got some pilot project funding from the SCORE and Howard Hughes funding. And then my first extramural grants in 2003 came from DoD and a NIH K08 award. The DoD award was focused on apoptotic regulatory changes in a molecule called TRAIL. The K08 award was on stromal epithelial interaction in the prostate. So those were sort of early, sort of nice, sort of confidence boosting grants that I received. But, this is just to show you that I've had a lot of unfunded grants over the years. So this is just one folder. This is about 4 or 5 years old now, this unfunded folder. There's a few more to add to this folder. But if you sort of count these folders, there are 102 grants that have been submitted and unfunded. So there's a lot that goes into sort of the 1 or 2 grants that are funded. You sort of take the victory and you're happy, you're excited, but there's a lot of downside to it as well. And I think you have to sort of be persistent and persevere and keep going. And hopefully, if you have the environment, as I did, with sort of the solid support of leadership that you guys here as well, I think it's a wonderful area to explore, because the type of gratification, satisfaction that you get from getting grants funded, from sort of scientific discoveries and moving the field forward, is sort of long term, and it's sort of a different type of a satisfaction than we get when we see patients on a day-to-day basis. We operate on them, they do well, they're very happy about their outcomes. So it's just sort of a working different part of your brain. So, everything was great and hunky-dory, but, early on in my career, my wife's a general surgeon, we had 2 kids, then I was submitting grants and nothing was getting funded. There was a lot of pressure to sort of see patients, and I was feeling like, Homer Simpson, feeling crazy and a lot of stress, both at home, both at work. And it can be quite challenging. And I think these are cycles that I think everyone goes through, as not many people talk about it. But I think it's cycles that people go through, particularly as your family gets more complex. You have kids, you may have a spouse. So these are things to keep in mind, that you're not alone in these types of challenges, both at home and at work. So I submitted a lot of grants to sort of get to the next level of grants of getting independent funding from the KOA and the DoD grants that I had and the institutional funding, and I couldn't get anything funded. I would submit 7 grants in 1 year and nothing would even get reviewed. So it was very sort of disappointing, until I came across this publication from NIDDK. They said, they're looking for applications that focus on genetics and epigenetics, and examining the epigenetic changes in men with BPH. And I said, I may not know a lot, but I know a little more than most urologists about epigenetic changes, because of the background and the experience that I had, and I could sort of apply it to urologically based diseases. So I sort of pivoted a little bit in my sort of research applications, and decided to sort of look at epigenetics related to prostatic cell survival and how it may affect the BPH, but it can also have effect on prostatic progression in prostate cancer as well. So I made a little pivot in my grant, because I knew a little bit about somatic inheritance of acquired traits and changes, like shown here. But epigenetic changes can be affected by aging, nutrition, environment. And these things can promote the cells to sort of alter their morphology, alter their underlying genetics, and then develop cancer cells that may be resistant. So I use sort of my background knowledge to apply it to a different type of a disease, which is obviously very important in urologic-based diseases as well. So I started submitting grants related to NIDDK, and I was happy to see that actually my grant was actually reviewed for the first time. So it still wasn't funded, but it was actually made it to the table and it was reviewed. So it got a score of 34 and a 38th percentile. So this sort of gave me some hope that things that there may be a light at the end of the tunnel. We worked this grant and then we resubmitted it, and luckily got a much better score, third percentile score that was funded. And this was after 10 years of being a faculty member. So it takes time. It takes a lot of grit and perseverance to sort of get these grants funded. But I think once you do, and hopefully if you're in a supportive environment, I think it's extremely gratifying. So what's our agenda today? So let's get to the scientific part of the talk. We're going to talk about 5-alpha reductase 2, also known as steroid 5-alpha reductase. SRD5A2 is the nomenclature that I'm going to be referring to it throughout the talk, and how it affects BPH. So we're going to look at epigenetic regulation of 5-alpha reductase 2, how obesity, inflammation, and prostatic regulations are sort of intermixed together. And then I'm going to sort of towards the latter part of the talk, talk about alternative androgen-independent pathways of estrogen-mediated and Wnt-mediated pathways. And we're going to finish the talk by what are some of these clinical implications of how we are in the process of developing or studying novel, non-invasive biomarkers and new treatment strategies related to sort of the earlier part of the talk. So prevalence of BPH, obviously it's well known to this audience that it's increases with age. The Baltimore longitudinal study showed that about every year, about 2.5% growth in the prostate, very common in elderly men. More than 50% by age 60, 90% by age 80 are affected. And at any one time, 6% of the world population is affected by some type of a BPH-related lower urinary tract symptoms. So shown here are CT scans of two different individuals, similarly aged, similar comorbidities. But as you see here, the individual on the left-hand side of the image has a very small prostate, about 40-gram prostate, whereas the one on the right side of the image has a 300-gram prostate, urinary retention with an indwelling Foley catheter in the middle, with urinary retention. So prostate is actually the only solid organ that grows during the adulthood. And as a result, BPH should be considered the most common tumor worldwide. And that's not how we think of it as a tumor, but it is a tumor, but it's just a benign tumor that grows. The global implication of BPH is huge. Just in the United States, it's $4 billion spent on management of BPH alone. The disability-adjusted life years for a lot that are lost due to BPH is very high globally, much more in the developing countries than the developed countries, but it significantly affects the world globally. And actually, there's a lot more deaths secondary to secondary complications of BPH cause more deaths globally than prostate cancer does. So it's a significant health-related issues both here in the United States and across the world. And we've heard that it does affect the royal family as well. So King Charles was obviously affected by it and had to have surgery for his prostate. And it not only affects the royal family, but also affects the royalty of the Hollywood area as well. So I'm going to just, if you don't know, this is Larry David. He's the creator of Seinfeld, a popular sitcom show. If some of the younger residents may not know about Seinfeld, it's worth looking it up on. I think it's showing on Paramount Plus. My son, who's 24 years old, he loves it. So he thinks like he's watched every single episode of it. And then now in this show, he has created the new show called Curb Your Enthusiasm on HBO. And so let's see what Larry David has to say about his concerns with his urinary symptoms. Oh, hold on one second. Stacey, can you help me with the volume here? So anyways, that was meant to show that he's spending a lot of time in the bathroom trying to make the best use of his time, learning about the Gettysburg Address and sort of reciting it. And so our goal basically is to sort of define mechanisms of resistance to BPH related medical therapy. And to achieve this goal, we have focused our attention on 5-alpha reductase 2, which is a key enzyme for prostate development and growth. And we're all aware that the drug finasteride is a competitive inhibitor of 5-alpha reductase 2, which converts the testosterone to the more potent androgen dihydrotestosterone. Commonly used for management of BPH, is actually the second most common prescribed drug by urologists after tamsulosin. So despite its common use, we know that about 36% of men, based on the MTOP study, are resistant to this medication. And sort of before we sort of go on further, I think it's worth sort of looking at the steroidogenesis pathway, and gives us an appreciation of how all of these sort of cholesterol-based compounds are linked together. So maybe I'll sort of reach out to one of the residents. Is Stephanie here? Stephanie Hanchuk?
Yes, hi.
Hey, how are you?
Good, how are you?
I was really impressed by your website. You have, like, all the residents, you have your own website, you have your own bio on there, you have your plans, so it's very well done. And I was extremely impressed at the fact that you actually skipped kindergarten. And so you must be very smart. So
No, I don't share, I can't color the lines, still working on tying my shoes.
So can you tell me, and you can sort of call a friend if you want, but maybe your colleague, co-chief resident who's going to pediatric urology, if you choose to, what's the most common sort of genetic abnormality associated to the steroidogenesis pathway for new babies that are born?
I know, isn't it these 17 alpha
You're close,
hydroxylase?
you're close.
Then 21.
21 hydroxylase deficiency, exactly. And the way that you measure it is by upregulation of 17 alpha hydroxy progesterone. So if that's upregulated, so every child that's born in the world is actually checked for this congenital abnormality, because about 1 in 500 to 1 in 1,000 individuals are affected by this deficiency. So Ben, you're going to pediatric urology. So what's the cause or that sort of sequelae if a baby has this abnormality? Yes, perfect. You're going to do great at Emory. So so salt wasting crisis, and 95% of individuals with congenital adrenal hyperplasia have this deficiency, which leads to hyponatremia, metabolic acidosis, hyperkalemia, and hypoglycemia. And there can be also some degree of virilization depending on how severe this form is. So for some of the younger trainees, basically when the 21 hydroxylase is this pathway is blocked, you're not having any of the aldosterone, corticosteroid, or cortisol levels produced, or any of the mineralocorticoids, and that's why it leads to salt wasting crisis. The reason you may end up, or the baby may end up with virilization, is because a lot of the sort of the byproducts of this pathway is pushed this direction towards the androgens and the estrogens, and that's why different degrees of virilization occurs. So what we're going to sort of talk about during the talk is actually more focused sort of in the bottom half of the slide on 5-alpha reductase and aromatase compounds that are responsible for testosterone and estrogen regulation. And we're going to focus on this aspect of regulation of 5-alpha reductase 2 during the talk. This other 11 beta hydroxylase, which is highlighted here, is just a sort of a lesser form of congenital adrenal hyperplasia, which I won't talk about much. So congenital absence of 5-alpha reductase 2. Amir, can you tell me do you know what happens if there's a congenital absence of 5-alpha reductase 2? Is Amir here?
He's absent.
He's absent. Okay. Ankur, can you tell us?
Maybe on Zoom.
So there could be, feminization, that's good. So what happens is that the male-born children can look like this, right? So what happens as they sort of get closer to puberty or they age, it's discovered that they actually have testes in their inguinal canals. And can anyone sort of tell me who had this discovery and how was this discovered, this congenital absence of 5-alpha reductase 2? It's one of the common, tidbits. It's funny, the older we get, sort of the residents know less and less of these things. So it was actually discovered in a Dominican Republic family, and it was discovered that these individuals lived a normal life. When they got older, they basically had really small prostates, and they had no urinary tract symptoms. So this is how this genetic defect was discovered. Was published in the New England Journal of Medicine and Science by a group at UT Southwestern in Dallas and a group at Cornell University. Does anyone know who was the author, or who was the discoverer of the New England Journal publication? Someone prominent in our urology field.
Goodman and Gilman at Southwest.
Yeah, but the first author of it was actually Dr. Pat Walsh. He's one of the prominent. Do the residents of Dr. Walsh is? Okay. Do you? And many of the residents actually don't. So he's one of the sort of the pioneers in our field. He, in addition to many of the scientific discoveries, he was the one who pioneered sort of the nerve-sparing radical. We call it anatomic radical retrograde prostatectomy. So a lot of contributions. In many different areas, both scientific and surgical. So this a little bit of fact of what happens when congenitally 5-alpha reductase 2 is absent. And we've also learned, so as a result of this congenital absence of, let me go back, of 5-alpha reductase 2, a drug was created called finasteride. So it didn't really help the family who had these congenital defects. But because of these clinical features of small prostate and lower urinary tract symptoms during adulthood, this drug was created that was called finasteride, which led to publication of the MTOP study, which is the medical therapy of prostatic diseases. And what they showed is that with use of finasteride, there's about a 20%, 25% reduction in total prostate volume. However, these symptoms progressed in about a third of the individuals, despite taking the finasteride. And that's what's sort of shown here in the MTOP study publications from New England Journal of Medicine by Dr. McConnell and colleagues. Also one of the best sort of well-done randomized clinical studies in the urology field. So if you're not familiar with it, I know Dr. Singh is in charge of your journal club. I'm sure he's going to bring that up for you guys so that you're aware. So, and then we've also shown ourselves that if you stay adherent to finasteride, it has a protective role in sort of averting the need for BPH-related surgeries. So if the patients stick with it, they have the best long-term outcome. But despite that, 30% are resistant to the medical management, and there's still a 10% annual incidence of progression of the urinary symptoms despite its use. So our primary goal has become to define mechanisms of resistance to 5-alpha reductase 2. And the way that our lab is focused on this is to determine the mechanism of resistance. We focus on molecular aspects, cellular and clinical aspects by looking at epigenetics and factors associated with 5-alpha reductase 2 changes. Cellularly, we look at heterogeneous cells population within the prostate, whether it's human or animal cells. And clinically, we're looking at variable growth rate and growth patterns of different zones of the prostate and how we can translate those findings to relevant clinical biomarkers and new treatment strategies, which we will finish at the end of the talk. So when we started this journey about 15 years ago, what we found was that there are actually epigenetic changes that affect gene expression of 5-alpha-reductase 2. We demonstrated that the expression of 5-alpha-reductase 2 is variable in different adult individuals. What you see in the top left-hand corner here is an individual which has a lot of 5-alpha-reductase 2 expressed, demonstrated by the brown immunoreactivity stain that you see. And in another individual, you see very little expression. In the immunofluorescence at the bottom, the green represents 5-alpha-reductase 2. You see one individual who has a lot of expression, another who does not. And then we define the mechanism that affects this variable expression amongst different individuals. It's shown here. Basically, if you have methylation of the promoter region, so this represents the gene 5-alpha-reductase 2, this is the promoter. And if the promoter is methylated here, through epigenetic regulation or epigenetic modification that I touched upon, this silences the gene and it doesn't transcribe the gene, and then the translation to a protein is not produced. This is just to show a single example of two different individuals at the sort of the very, drilled down genetic level. The closed squares, the black squares represent an individual that has methylation of the 5-alpha-reductase 2, and you see in another individual, the open squares demonstrates no methylation. So there's significant variability in different individuals of how the promoter is methylated. So we defined over the years the pathway that regulates the methylation of 5-alpha-reductase 2 is through inflammatory mediated pathways. So that's the main thing you need to remember, that inflammatory mediated pathways can affect methylation and also the expression of 5-alpha-reductase 2 during adulthood. And then we asked whether aging and inflammation can affect the expression, and this is like the only slide that's going to have some gels in it. But we saw in prostate of humans that TNF-alpha in older individuals, there was much higher level of inflammation of TNF-alpha and IL-6 as compared to younger individuals. And if we look at animals, so now these are mice, wild type, genetically normal mice. As the mice age from 3, 6 to 12 months, there can be some mice that lose expression of 5-alpha-reductase 2 in their prostate. And then the genes that we had shown that regulate 5-alpha-reductase 2, the NMT1, can also be upregulated, which promotes the methylation of these genes. And then the methylation activity can actually increase of the promoter region with age. So the older the mouse, more methylation activity of 5-alpha-reductase 2 and more activity of the genes that regulate the methylation of 5-alpha-reductase 2. So a resident at MGH, Seth Beaches, who is now a faculty member, a young faculty member at UC San Diego, demonstrated that in our clinical cohort, age and the body mass index predict 5-alpha-reductase 2 methylation. And as you see, both with age and body mass index, the red curve moves to the left-hand side, demonstrating a higher positive rate of 5-alpha-reductase 2 methylation. And all of the clinical parameters that we looked at, it was only BMI and age that sort of came out as in the multivariate analysis as being significant, suggesting that both older age and higher body mass index can affect as an environmental factor the epigenetic regulation of 5-alpha-reductase 2. So here we're looking at, again, patients with different body mass indices, which demonstrates here that with a higher body mass index, the percent of methylation increases in the promoter region, as shown here, and the expression of the gene decreases. So higher methylation, lower expression. So more epigenetic sort of modification of the gene, lower expression of 5-alpha-reductase 2. And if we sort of look at compare the inflammatory changes both in mice and humans, the mice is in the top part of the graph, human in the bottom, the different factors of inflammatory mediated factors are upregulated. So in mice, we give them high-fat diet, as shown here, and all of these inflammatory mediators, the IL, the interleukins, and TNF-alpha are increased. And individuals and in humans with the higher body mass indices, similarly in the prostate, the inflammatory mediators are increased. So Rod Alfaori, who has been working with us for the last 2.5 years, and is going to actually be joining us as one of our incoming residents, asked a question from our cohort of patients at Beth Deaconess Medical Center. Do anti-inflammatory mediators in individuals who may be on anti-inflammatory mediators for various conditions, arthritis or various issues with colitis, do they tend to have smaller size prostates if we follow them over time? So he queried over 1 million patients in our data system and sort of narrowed it all down. And basically, the take-home message of the study that's impressed, what he showed is that if the individuals have had no anti-inflammatory mediated treatment, their prostate size tends to be larger over time than individuals who have been treated with TNF-alpha inhibitors. And the growth pattern over many years tends to be slower if the individuals have been treated with TNF-alpha inhibitors. Again, another way suggesting that inflammatory mediators can affect prostatic growth, prostatic size over time. And just another way of demonstrating the two links between prostate and inflammatory mediators. So here's a question. Expression of 5-alpha reductase 2, also known as SRD5A2, is similar in all adults. Is Christopher Hayden, are you here? What do you think?
It's false.
Perfect, good. So I think Say is here, right? How about this one? Methylation at the promoter region of the genes leads to and select the best answer. So that's a
Decreased expression of the genes.
would you say increase or decrease?
Decrease.
Decrease. Perfect. You guys are all over it. Excellent. Wonderful. So and then is Soam here? Lecture. I was impressed. He said he's a brunch enthusiast. I don't know what that is.
Like to eat.
How about Say here?
Yeah.
Oh, I'm sorry. Yes, I asked you already. Dylan, is Dylan here?
Just stepped up.
Just stepped up.
I mean, back.
Okay.
Dylan's back.
This is.
Oh, there you are. Okay. Yes, we met earlier. Yes.
Oh, God.
So what do you think? So A and C. So we have increased inflammation and decreased expression of 5-alpha reductase 2. So with aging, you get more inflammation, and as a result of the inflammation, it promotes epigenetic regulation and decreased expression of 5-alpha reductase 2.
I have larger prostates than older patients.
Well, it's still so we'll get to that. So remember alternative pathways, and we'll get to this. Excellent. So in summary, so this first part of the talk, we've talked that inflammation, age, and body mass index are associated with 5-alpha reductase 2 promoter methylation. Patients with BMI greater than 25 have higher methylation levels and lower protein expression. And then elevated inflammatory mediators in prostatic tissue are associated with obesity, and inhibitors, TNF-alpha inhibitors or anti-inflammatory mediators reduce total prostate size and growth rate. So we've known over many years that in addition to sort of androgen-mediated pathway, estrogen-mediated pathways also regulate prostatic growth. And this is just a quick summary of a review paper that shows how stromal epithelial interactions and also the estrogen-mediated pathways can affect prostatic budding and prostatic development. And some of the molecules that are shown here, like the Wnt5A pathways, have been suggested to be mediated through estrogenic-mediated pathways. So because of this, we looked at sort of gene expression of individuals who have 5-alpha reductase 2 expressed and those that who didn't. So on the left-hand side of this gene expression profiling pathway gene expression profile are individuals without methylation. On the right-hand side are those with methylation. And then what we found was that 3 classes of genes were upregulated between the two sort of comparison of the 2 cohorts. But the area that we sort of decided to focus on and was most exciting to us by this initial analysis were the estrogen response genes that were upregulated in individuals that had 5-alpha reductase 2 methylated. So what we found, we looked at individuals' prostate tissue and those that had 5-alpha reductase 2 methylated. So we showed that with methylation, degree of 5-alpha reductase 2 expression is lower. As expected, the level of testosterone is higher in the group. Dihydrotestosterone level is lower in the prostate, but the sort of the surprising finding was that the estrogen levels were actually also higher. So this was sort of a new finding for us back in 2017, that the estrogen levels are also upregulated, and this had not been reported in any of the other studies in the past that have looked at these. And here, what I'm showing you is that in absence of 5-alpha reductase 2, on the top is an individual with 5-alpha reductase 2 expressed. The bottom, an individual does not have it expressed. And the area to focus on is aromatase, the enzyme that converts testosterone to estradiol, and an estrogen receptor alpha, or phosphorylated form of estrogen receptor alpha, which is significantly upregulated here by the brown staining, as you see in the individual that does not have 5-alpha reductase 2 expressed. So we went on to sort of evaluate this a little bit deeper. In mice, we looked at mice that had high fat diet exposure. So with obesity, estrogen-mediated pathways shown here by the CYP19A1 is upregulated. Similarly, in humans with body mass index above 25, we had higher levels of this estrogen-mediated compound, and aromatase was also significantly higher in the condition media of the prostatic tissue as well. All of which sort of demonstrate that there are alternate pathways when 5-alpha reductase 2 is absent. So what we coined back several years ago is that in the absence of 5-alpha reductase 2, there's an androgenic to estrogenic switch. So testosterone to dihydrotestosterone with 5-alpha reductase 2, when it's blocked in 30% of individuals, as we've shown, it leads to upregulation of the phosphorylated form of estrogen receptor alpha. Aromatase enzyme that converts testosterone to estradiol is upregulated, and we get upregulation of estradiol. And all of these are mediated by inflammatory mediators on both ends by TNF alpha that we've shown through our publications. And this androgenic to estrogenic switch, I think, brings us opportunities for novel and targeted therapies. And we published this back in 2017. So we developed this hypothesis that non-androgenic pathways are activated and maintain prostatic development and growth when 5-alpha reductase is suppressed. So we developed this hypothesis. We developed this mouse model where 5-alpha reductase 2 is absent. So 5-alpha reductase 2 knockout mice. I'm not going to go through all of the studies to show that 5-alpha reductase 2 is actually knocked out, but take my word that it is. And then in the mice, what we see here is that basically the prostate and the seminal vesicle for the mouse that have genetically knocked out 5-alpha reductase 2, they're smaller. Basically, this shows that in these mice, the urogenital weight to body weight is smaller, and the prostate to body weight is also smaller in the knockout mouse. And the relative expression of 5-alpha reductase 2 is basically very, very minimal in the knockout mouse in their prostate tissue, showing that what we created is what we expected. And then in these mice, however, what was surprising, we found that there is lower level of apoptosis and higher level of cell proliferation. So in these genes here, these are upregulated apoptotic mediated genes in the knockout mouse. And then when we look at the cell proliferation, it's actually higher in the knockout mouse than the wild type mice or the heterozygous mice in this case. And this was surprising to us. So this tells us that despite the fact that the body weight is lower, there's more sort of activity in maintaining the cell's growth pathways. So what we did to sort of identify sort of what are the population of cells that are growing, we did a single cell RNA sequence analysis. This shows all the different types of cell populations within the prostate. And this shows all of the different types of epithelial cells. Specifically, there are 5 different subpopulations of epithelial prostates, LE1 through LE5, in the mouse prostate that we looked at. And the one that particularly sort of stood out to us were these LE2 cells. So compared to all of the other cells, the LE2 cells were increased in the population of cells in the mouse prostate. So we focused our attention on these LE2 cells because there were more of them in the knockout mouse, and we saw also more proliferation and less apoptosis in the knockout mouse to see are these, and that question is, are these the cells that are maintaining the growth of the prostate in these knockout mouse? So what we found was that in the LE2 cells population here, that again, we come back to estrogen-mediated pathways, this ESR1, or which is also the estrogen receptor alpha, is significantly upregulated in these LE2 cells. And the LE2 cells that are shown here, that have these genes that are specifically upregulated in these LE2 cells that also have sort of higher proliferation and less apoptotic rates. And the pathways that are involved with these markers are MAP kinase pathways and Wnt signaling pathways that are upregulated in this single cell RNA sequencing analyses. And where do these LE cells, where are they localized? They're mostly localized in the anterior prostate lobe. There are 5 different lobes in the mouse prostate, so they're localized in the anterior prostate lobe. And if we look at the markers that are upregulated, we see that these markers by RNA FISH analysis, this ESR1 and PPT1 that are markers for the LE2 cells are more upregulated in the knockout mouse here than the wild type and the heterozygous mouse. So, and in the humans, well, how does that translate to the humans? So what we found in individuals that express versus those that do not express 5-alpha reductase 2, we see more of these markers of the LE2 cells. So when 5-alpha reductase 2 immunoreactivity score is decreased, we see more and more of these LE2-like cells in the human, similar to what we saw in the mouse prostate. And then this is looking at the ligand receptor interactions, and all I'll just leave you with is that there are the Wnt-mediated pathways. When 5-alpha reductase 2 is absent, you see with these sort of increased circles, higher expression of these ligand receptor interactions with the Wnt signaling. And then Christina Sharkey, who's been with me for several years as a research assistant and is now a graduate student at Northeastern University working with us jointly in the lab, showed that in the knockout mice, the Wnt signaling pathway is significantly increased in the knockout mouse when 5-alpha reductase 2 is absent, suggesting these alternate mediated pathways that maintain prostatic growth. So let's do one more question. Is Jeff here?
Yes.
Jeff, can you take that question? So in the absence of 5-alpha reductase 2, perfect, nice. So we have increased testosterone that we already know. There's decreased DHT. There's a shift to estrogenic pathways as alternative pathways. So all of the above.
All right, that's
Yeah. Yes.
so it's so interesting, fantastic. You know, when you block 5-alpha reductase 2, you get increased testosterone, you get shifting to estrogen, especially if you're obese, more aromatase activity. But in your model, it's an early knockout, so you're going to affect the prostate development
At the early stage, you're right. Yeah, so it's not a BPH model, it's looking at a survival model of the cells.
right? Because if you had an induction model, then the prostate would develop normally, then you could.
Right, so that would require a sort of a model where we can turn on and off, like with tamoxifen or something like that. So that would be sort of our next step of
Or
an
androgen
animal
receptor
model
block.
that we have to.
Yeah.
So, but that's a good question, but the model that we have is not looking at a BPH model, but a survival and persistence of various stuff there. So we get that criticism on many of our grants. So thank you for bringing that up. So that's perfect. That's an excellent question. So in summary, so I know my time is running short, so I want to get to sort of the clinical aspects of the talk. So we generated a 5-alpha reductase 2 null mouse model that only suppresses generation of DHT but maintains activity of testosterone. We defined a discrete subpopulation, specifically the LE2 subpopulation of cells in the mouse prostate from the anterior prostate that has increased expression of estrogen of estrogen receptor alpha and ESR1 in the absence of dihydrotestosterone. And then in the absence of 5-alpha reductase 2, there's a stromal epithelial interaction, ligand receptor interaction, where the Wnt signaling pathway could potentially work in promoting cell survival and persistence as alternate pathways. So what we did to sort of bring it to a clinical relevance, we got tissue samples from the MTOP study. So this was like 5,000 patients who were treated in a randomized trial with finasteride in sort of the late 1990s, early 2000s. And we had the fortune of getting our grant accepted by NIDDK so that we could ask clinical questions of identifying good responders to finasteride versus poor responders to finasteride. And then try to ask the question, is there a difference in expression of 5-alpha reductase 2 between individuals who responded well versus those who did not respond well? So at the top you see here is an example of a good responder. And basically you see a lot of immunoreactivity, a lot of brown staining of 5-alpha reductase 2. In contrast, a poor responder at the bottom shows very little to no expression of 5-alpha reductase 2 by protein expression. So, and then we looked at the expression over the 5-year overall response by the AUA urinary symptom score, and you see a direct negative correlation between expression of 5-alpha reductase 2 in the x-axis and the improvement score. So the lower score obviously is more improvement. So the more expression of 5-alpha reductase 2 is associated with better response to finasteride therapy from the MTOPS study samples. So translating our studies from bench to bedside, we want to ask, can we use this 5-alpha reductase 2 expression and methylation as a biomarker for someone who may or may not respond to the drug? We don't want to do a prostate biopsy on every individual that we are we're prescribing finasteride. So we asked the question, can the methylation result from the prostate also mirror the methylation of 5-alpha reductase 2 in white blood cells, in circulating white blood cells? So is there any direct correlation? So this first graph, what I'm showing you is a pilot study that we did looking at expression of 5-alpha reductase 2 and in the prostate, and also methylation. And as we've shown before, there's a negative correlation. So more methylation, less expression of the protein. And then what was sort of surprising and sort of novel to us was that when we looked at white blood cell methylation, there was a direct correlation with prostatic methylation of 5-alpha reductase 2. So we could potentially use this as a biomarker by basically taking peripheral blood sample and have an assay where we can evaluate methylation of 5-alpha reductase 2 to basically use this as a finasteride response predictor. So we take white blood cells here, we assess for methylation of 5-alpha reductase 2, and how we do that methylation analysis, we could analyze the whole promoter region, we could analyze some parts of it, there are parts of it that are more methylated than others, and those are things that we hope to sort of figure out in the next stage of our studies with a new grant that we just submitted recently. So having a non-invasive biomarker. As you recall, I also told you that inflammatory mediators are also important in regulating this epigenetic modification of 5-alpha reductase 2. So in another sort of bench to bedside analysis, we use this nanoparticle pheromoxetol enhanced MRI that is a marker for inflammation in the body. So this inflammatory marker using an MRI study, what we saw here are two different individuals with prostate MRIs. And in the top, you see a significant T2 signal drop, meaning that there's a high amount of inflammatory cells, macrophages specifically in the prostate of this individual, whereas in this individual, there's not a significant change in the pre or post infusion of this nanoparticle ferumoxytol. So this individual has a lot of inflammatory cells, this individual does not in the prostate. And then we asked, does this signal, in this pilot study, correlate with methylation of the transition zone of the prostate? And there's a direct correlation. So there's more inflammation, more macrophages on the prostate, and then there's more methylation of the 5-alpha reductase 2 tissue from the transition zone. And again, the expression of 5-alpha reductase 2, there's negative correlation with the tissue methylation. And basically, so higher nanoparticle ferumoxytol uptake is associated with higher methylation of 5-alpha reductase 2 and lower protein expression. So as a result of this, this is yet another non-invasive pathway that we could use to use the inflammatory mediated pathways as a signal to see who may or may not have epigenetic modification of 5-alpha reductase 2 and potentially as someone who may or may not respond to the drug. So our approach in our sort of studies moving forward is to define non-invasive biomarkers. One is through white blood cells to get a methylation score of the promoter region, and another using MRI and an inflammatory signal using a nanoparticle to get an MRI score to see whether that correlates with responsiveness to the drug. So in conclusion, what I've shared with you is expression of 5-alpha reductase 2 is variable in the adult prostate tissue. Epigenetic modifications, specifically methylation, inflammation, environmental factors like body mass index and age can affect expression of 5-alpha reductase 2. With decreased 5-alpha reductase 2, there's an androgenic to estrogenic switch. With increased 5-alpha reductase 2, individuals who have higher levels of expression, there's a better response to 5-alpha reductase inhibitors, as I showed you from the samples that we got from the MTOP study. And if you have lower levels of 5-alpha reductase 2, the patients tend to have a worse, poor response to the drug. And we're working on non-invasive biomarkers, blood and MRI, to predict sensitivity to 5-ARI therapies. So where are we going in the future for our future studies? We're proposing a clinical trial for new treatment strategies and epigenetic biomarker. And what we want to do is to assess the role of combination therapy. So both the androgenic pathway and the estrogenic mediated pathway for management of BPA. So we postulate that in individuals who have 5-alpha reductase 2 absent, combination of finasteride plus a SERM, an estrogen modifier like raloxifene could potentially work better. And that's the clinical trial that we're proposing. And hopefully NIH will enable us to carry out that clinical trial. We want to prospectively determine the role of methylation of 5-alpha reductase 2 from peripheral blood to see if we can use it as a biomarker, and then to prospectively also evaluate the radiologic marker, the inflammatory marker with the nanoparticle ferumoxytol to see if that can be used as a predictor for promoter methylation of 5-alpha reductase 2 and as a marker for response to therapy. So with that, I'll end. I'm thankful to individuals in my lab who have been instrumental in our studies over the years, our collaborators in radiology, pathology, our colleagues at UT Southwestern. And I want to put a plug for this program that we have at Beth Deaconess Medical Center, the FIRST program, which stands for Facilitating Innovative Research and Surgical Trials. That is truly a sort of an outstanding model for any type of a clinical or translational studies that they support us in our biostatistics, grant writing, IRB protocols, and so forth like that. So without them, I think that these trials would have been much more challenging. And I wanted to just give a plug for Zhongwei Wang, who's been with me for the last 15 years, and he's been the one helping me with all of our studies. And finally, I'll just end it with a picture of my family on a sailing excursion that we were a couple of years ago. So thank you for your time. It's been a wonderful time being with you, meeting the residents, spending time with the attendings. And you guys have a wonderful program. You should be extremely proud of where you are, and you have all of the tools available to you to do whatever you want to do. So thank you.